Okay, let's get back together, please. Let's talk about just a little bit about this topic here, the evolutionary timeline. Okay. Some students find it surprising or interesting that the first organism that ever lived on Earth was a bacterial cell. So bacteria predate us for quite a while. They predated us. If you look at the timeline, we are recent arrivals. When you take into consideration the age and how old Earth is, we're here, recent arrivals. So what was going on? It was actually a bacterial world for billions of years before we ever existed. I'm talking about the eukaryotes even, not just us. Eukaryotes ever existed. So the theory is that we're all descendants of an ancestral cell. Long time ago, the ancestral cell adapted, evolved, and produced bacteria, then archaea, and then the eukaryotes. So the idea now is, or the question now would be, how did bacteria survive for so long? Thinking about all the environmental changes and everything, and they're really very they have ways of surviving. They know how to survive, adapt and survive. So how? Well, one reason is because of their ubiquity. And that's what you set up 2 days ago. When you were sampling the countertop, remember that? And then opened the plate to the air. Remember that, hopefully? You just did it 2 days ago. And then your fingertip and your coughing and your hair scratch. What were you testing? Nobody asked me, what are we testing? We're testing, are bacteria ubiquitous? That's what we're testing. The word ubiquity means everywhere. They are all around you. Yes, they are. They're all around you, they're on you, and they're in you. The rough estimate is that you are made up of around 10 trillion cells. 10 trillion, that's a lot of cells that make you who you are. But the bacteria that's in you and on you, they add up to about 100 trillion cells. They're 10 times more in number than you are. So besides the fact that they are ubiquitous, which means they're everywhere, they're in the air, they're on my desktop, they're on my finger, they're in my coughing and my hair scratch. That's what we'll see on Tuesday when we look at your plates. They're also abundant. There's plenty of them. So do not confuse ubiquitous with abundance. Ubiquitous means everywhere. Abundant means a lot of them. That's the difference between. And the main reason, besides the fact that they're everywhere and they're abundant, one of obviously the main reasons would be is they have a very easy way to adapt to changing environments. And they have adapted them along with the archaea to environments that no way you and I would be able to survive there. There's no way. In places on Earth that you would not even dare go to because of how cold it is, and if you're caught for about 2 or 3 minutes out there in that cold, you run the risk of dying. Really fast. They're surviving there. In depths in the ocean, where there's no way you can survive down there without being in a protective vehicle because of the pressures. As you dive deeper and deeper and deeper in the water, I don't know if you know this, but pressure increases. You're going to get squished like a can. Like that. Well, obviously you can't do that unless you are in a some kind of vehicle that is pressurized. Otherwise you won't survive that. But bacteria, some of them at least, and the archaea, they live there. That's their home. That's what they adapted to. You know, if you take some of them and you say, I'll save you, I'll bring you up to the surface, they die there. The pressure is too low for them, and they won't survive. Salt. There are some environments that are extremely salty. They survive there. You name it. There is some bacteria, and especially archaea, that live there. Because archaea are collectively referred to as extremophiles. Extremophile. That is a combination of the word extreme and the word phile, means loving. Loving extreme conditions. I am not an extremophile. No, I don't like that. But these archaea, they love it. They love high salt, some of them. High pressure, some of them. High temperature, some of them. You name it. Low pH, high pH. So collectively we call them extremo, for the word extreme conditions. Phile, loving extreme conditions. Now there are some bacteria that do the same thing, but I'm talking about archaea. Archaea are professionals. Professional extremophiles. So these are the key factors why bacteria survive for all this time. The other thing is their essential role in life. Because they're essential, you want them to hang around. You know, if we go on the attack, if we decide as a group, hey, let's get rid of all of them, meaning bacteria, you know who's going to die shortly after that? All of us. You can't do that. Bacteria does a lot of things for you and I. I know there are some that are bad. We call them pathogenic.
Cause disease.
Yeah, the ones that cause disease are referred to as pathogenic, disease-causing. Those you want to stay away from. But most of them, let's say for example 90% of them, are non-pathogenic. You shouldn't be afraid of them. They actually help you. They help you, like we said, digest our food, keep our immune system in check, strong. Do you know that your gut bacteria, nowadays we found out that our gut bacteria has an effect on our brain function? How? We'll talk about it. Bacteria in the environment has another huge role. Huge. They recycle things.
Yeah.
When an apple falls from a tree, don't pick it up. Just leave it alone. Maybe 2, 3, 4 weeks later, it won't be there anymore. Why? It decomposes. It gets broken down and recycled back into the earth. Who's doing that recycling thing? Bacteria and fungi. Those are the two main recyclers. Bacteria and fungi. That's a nice question on the exam. There you go. And there's a word for that. We refer to them as saprobes. Saprobes. microbes. Those are the organisms involved in decomposing dead matter or organisms for the sole purpose of recycling. What happens if they don't recycle? We're gonna run out of things like carbon and nitrogen. We'll simply run out of resources. After a long life to you and I, we are no different than that apple falling off the tree. No different.
Absolutely.
The fungi and the bacteria that live within you and on you, they're already there, will also recycle you over a long period of time, but it's inevitable. It will also recycle you. Now, there are some civilizations, the older civilizations, they were like, where I'm from, Egypt, they had this religious understanding or belief that they needed to preserve the body. So they had to do something. I mean, these guys, they're amazing. They knew about this stuff without knowing about the stuff. You get it?
Mm-hmm.
Do you think they had a microscope back then? As far as we know, they did not. But they knew there was something that if we don't do anything to the body, it will decompose over time. But their belief is this person is coming back and he needs his body. So we're talking about the pharaohs, right? So they figured out a way to preserve the body. So from my understanding is that they would put the body on the table and throw a whole bunch of salt on it. A mountain of salt on it, like that. So it's very simple. Put the body on the table, drop the salt. 3 weeks later, I'll come back. They come back 3 weeks later, they remove the salt, it's already mummified. Now, some people think that it took 5,000 years to mummify the body. No, it took 3 weeks. He looks like after 5,000 years. No, he looked like that after 3 weeks of this procedure. What did they do? They removed what? With the salt. What goes towards the salt? Gen Bio 1. Water. They sucked the water out of the body. Why? Because the bacterial enzymes, the bacteria, to decompose something, they use enzymes. And the same thing with fungi. They secrete enzymes to digest whatever that is. Well, for enzymes to work, they need water, hydrolysis. Do you remember that, Gen Bio? The break-in by water and an enzyme. So you're inactivating them. You're removing the critical thing that they need to keep their enzymes active, water. So even though this person had bacteria within it and on it, but the salt inactivates all of their enzymes. The only thing that you need to do now is make sure the water doesn't come back. At any given time, the water comes back, that body will disintegrate real fast. And that's why they wrapped them, right? Mummies. But the other good thing or benefit is the environment in Egypt. It's a dry environment. It's a desert, dry environment. So they didn't have to worry about wrapping them and encasing them and to prevent, I mean, if they do that here, good luck. It won't last that long. There's too much water in the environment here. But over there, unless you go to the Nevada desert or something, but over there, it's dry air. There's very little humidity. So you can keep that body preserved for quite a while. And we're talking about thousands of years.
So it's only salt that they use because
It's all they use is salt.
I'm from Egypt and I thought they used something else like.
It's called I can't remember the name of the salt, but it's basically mostly salt. But they do other things. They remove the organs and all that. They did do that. They had this belief of they have to remove the heart and put it in a jar. Remove the intestines, put it in a jar. As if the person, when they come back, they're like, you see what I mean? That's what they believe.
They're in different jars of coffee.
It's in different jars. Yeah. But I'm not sure. I'm sure they use other things. But I'm just saying, quick. On the main thing is the salt. But you cannot bring the water back. So you're from Cairo, Egypt?
Yeah, Cairo, yeah.
Did you ever go to the museum?
Yeah.
Did you
not,
see-
not the new one, the old
No,
one.
the old one. I went to the old one. Did you ever see Ramses
Yeah.
II?
Yeah. That
You
was
know who
awesome.
Ramses II is, you guys? He's the guy, the pharaoh that had a fight with Moses.
Mhm.
Yeah. We're talking about thousands
And he's still
of
there.
years ago. I went to see him. I didn't even know he was there, right?
Mhm.
And I was standing right in front of his. They had him in a glass case, right?
Mhm.
And I'm standing there, I'm like, who is this? He's a really tiny guy, and he is literally not even taller than this table right here. Am I right?
Mhm.
Literally balding,
Yeah.
standard balding pattern. Who's this guy? Looks like a very important guy, right? Everybody else is in the case somewhere else, and these guys are. Oh my God, Ramses II. This is the guy that had a fight or a thing with Moses, right? Free my people and all that stuff, right? Little tiny guy. You wouldn't think he would create that much trouble. Oh, he did. If I remember right, he had 67 wives,
Jesus.
150 children. And the main reason behind it is, again, because of Moses. The story is this. I don't know if you know the story of Moses. Moses kept telling him, free my people, and he resisted. So he cursed the land of Egypt. And there were multiple curses. One of the curses is the oldest son
Mhm.
will be killed, dead. We don't know what they died of, but the story is that they did. The oldest son, each child, the oldest son of each family of the pharaohs in that area died. One of them is his kid. He's the only one.
Mhm.
He only had one child. And that curse killed him. And that's when he decided, and he went to Moses, he told him, get out. I don't want you here. You're causing too much trouble. And then the story is he changed his mind and he ran after them. I don't know if you know all that story. It's a beautiful story,
Well,
actually.
I'm Jewish, so
Yeah.
it's the story of Passover.
Of course, Passover. And then the splitting of the Red Sea and all that stuff, right? The interesting thing is this, after this has been done, what happened with Ramses? He got so upset that his son died in the middle of all this. And he wanted to make sure that he has enough children in case Moses comes back. You see what I mean? That's a true story. So he had all these wives, had all these children to make sure that at least some of them survive. Here's the interesting story now, to finish this off. Fast forward many years later. Back in the early 1900s, there was a British guy named Carter. He went to Egypt. He's an Egyptologist. He studies Egypt. And he's the one that found King Tut's tomb. Great, awesome discovery. So he was digging and he found it and they dug some more. And then the problem is when he's digging, right, him and his team, he has to get rid of the sand that he just picked up, right? So he goes like this. He digs. His objective is to empty King Tut's tomb.
Mhm.
And he goes like this. He doesn't know where he's throwing it. He's just throwing it. Many years later, someone decided to dig over here where Carter threw all of the sand, right? They found the 150 children of Ramses II.
Hmm.
This guy Carter could have found two huge discoveries right in front of him. He just happened to, he didn't have any space for it, and so he threw it. He focused on the gold and all that. I understand, right? And eventually they found an underground city with all the rooms, and each room had one of Ramses' children. So what do they want to do? And I don't know if they did it yet or not. Egypt has certain laws and rules that they will prevent you from doing something, even though it would be a good idea to do it, but there are rules that, the digging, and
Yeah.
there are things that they discovered that the government says you're not touching it. Okay, can you do it? No. So they're there waiting for us to discover them, but they're blocking anyone from studying it. One of the things is what? The common sense thing to do is what? Find out what his first child died from. His first child is there. And that will tell us what the curse was about.
Yeah.
What's the nature of the curse?
I don't think it's a good idea.
I don't know. I think it's a good
They
idea.
don't even watch the movies.
I know. What I'm saying is, can we find, was it a fungus? Was it bacterial?
Bacteria, yeah.
What is it? So they have the corpse or the mummy of this child,
Oh, wow.
but they are waiting to test it. At least I don't think they tested it yet. But what a fascinating story. Amazing. History is unbelievable if you just keep reading and studying this stuff. So let's get back to this. This obviously shows you the reasons, the four main reasons why bacteria, listen, existed all this time. Obviously they continue to be here, am I right?
Mm-hmm.
And the theory is, or the prediction is, after we are long gone, they will still be here. Because they have adapted to this. All right. Today, we talked a little bit about this already. We can use microorganisms to get things done for us. But again, we can go back in the past, again, back at the time of the pharaohs and stuff like that. They actually used microorganisms to get things done without even them knowing that they even existed. Making bread, they've done it in the past and we do it today. That's yeast. There are other ways of making bread today, but in the past especially, of course, they used yeast. Molded bread. Today, when our bread molds, we throw it away. Back then, that would be considered sacrilegious. Molded bread was used as a means for medication. Again, they don't know why. They did not know why, but they knew that when you take this molded bread and you put it on the cut, let's say you cut yourself, you take the molded bread and put it on there, it prevents whatever that is. See, they didn't even know what infection is. It prevents whatever that is that would happen 2 or 3 days later. So what's in the molded bread that's preventing that? Now we know. What is that? Penicillin. There you go. Now, for us, we don't sleep on molded bread anymore, right? We just take the penicillin. But technically, I know antibiotics were first discovered in the early 1900s or so by Alexander Fleming. But technically, they already were using it without knowing that they were using antibiotics. And the best part, to make beer and wine. That's the best part, right? And we still continue to do that. Preferably Heineken. And okay, besides the point. All right, so we do other things. We make vitamins and antibiotics with these microorganisms. Watch this one. We treat water treatment plants, create biofuels, and God forbid, war. Hopefully that never happens. But there are some countries who weaponize these pathogens. I have no idea why, knowing that any form of use of any of this kind of stuff would be completely devastating to all of us. And I have no idea what benefit that would serve us there. But they have them weaponizing these pathogens. Things like anthrax and stuff like that. All right, let's go to this part right here. That's called bioremediation. The word remediate means to fix a problem. If you ever get a phone call from your son or daughter's school, God forbid, and the principal says, we need to remediate your son's behavior. That means we have to fix his behavior. But in this case, it's bioremediation. It's fixing a problem using biology. That's what that means. So things like oil spills. A long time ago, not too long actually, but a while ago, they used to actually try to trap the oil, suck the oil, clean the... It becomes a huge job when you have an oil spill. Very bad for the environment, of course, and things like that. Nowadays, you may see this, God forbid, but you may see this where there's this guy with a white suit on, like a biohazard kind of thing on, and he's spraying it like that. He's going like this. And you stand on the side and you go, he's so nice. He's washing the rocks. He's so nice. He's not washing anything. He's spraying them with bacteria. It's the bacteria that's going to get rid of the oil after a little while. But his job is to walk around, make sure he covers the area with bacteria, right? And let the bacteria go to work. So what are we doing? We're fixing a problem, the oil spill, using biology. That's called bioremediation. We do the same thing with sewage treatment. All the water that goes into our sewer system, that's recycled. But they don't just throw it in there, I hope. It goes through a whole bunch of things for it to get cleaned. One of the steps along the way is introducing bacteria, actually, that degrade these waste products, that break them down. Of course, on the other end of that water plant, there are testers and so forth to make sure before they put the water back into the recycler, which basically like lakes and rivers, that it's actually clean. So don't go getting grossed out and all that. They're usually very good at that. Plastic degradation, that's a huge problem, continues to be a huge problem. You know that plastic bottle of yours? If you decide, for whatever reason, man, you're too lazy to go to the recycler bin or whatever, you just throw it in the regular trash. It will eventually sit in the landfill. And it will out-survive you, your children, and your children's children. Maybe they're also their children. It will just sit there. Do nothing. Because it's very hard to break down, plastic. Very difficult to break down. But now we have those bacteria that can, thank God, break down these plastics. So now they can introduce that in landfills, right? To help speed up the breaking down of these plastics that end up in landfills. The better place for plastics is a recycling bin, so that they can recycle them for some other purpose. But now we have them as well. So these are the three main things that are a problem for us, that we resorted to biology to solve the issue. Oh, I know. You don't need to know any of this. Oh, thank God.
Thanks, God.
Thank God. But I have to give credit where credit is due. I will see if I can talk about at least some of them. Maybe not all of them. But you can read about it in the soft chalk.
Yeah.
It all starts with the scientific method around, 1621 and so forth. You don't need to know dates. You don't need to know names. We just have to owe respect to them, because without them, you wouldn't be in this room learning microbiology. One of the things is Robert Hooke, and shortly after that, Anthony van Leeuwenhoek, if I'm pronouncing his name properly. They were the first ones that really started looking into the microscopic world. It was, of course, suspected prior to them, but they're the ones that actually went ahead and developed crude lenses and so forth, and to the point where you see, look, I told you. Then we also respected Joseph Lister for many reasons, but one of the reasons is he was the one that produced the prototype compound microscope. The first microscope that's similar to the one that you're using. was produced by Joseph Lister. Followed by Louis Pasteur. This guy, every time you turn around the corner, you see his name. He did so many things. One of the things that he did is that he simply disproved something called spontaneous generation. Listen to what they used to believe, which would be a disaster for you. They used to believe that if you wait long enough, another Dr. Saad will appear over here.
What?
That's what it is, spontaneous generation. Dr. Saad will spontaneously appear over here, which means you'll have 2 exam ones. One from me and one from him. And then if we wait long enough, you'll see another Dr. Saad.
Jesus Christ.
Oh, Jesus Christ is right? Now you have 3 Dr. Saad.
3 exam 1.
Then no, exam 1 is over. 3 exam 2.
Oh, Lord.
You catching it? That's why I said that would be bad news for you. It doesn't happen. Of course it doesn't. Thank God. There's no such thing. After that, there was another thing that came after that, of course, something called the cell theory. Cells beget cells. You want a cell? Yeah, a cell comes from a cell. So you can't have the Dr. Saad just spontaneously appear. Thank God. So he disproved it. He said, you guys are crazy. That doesn't work that way. He actually had to do an experiment to prove to them that that's not what happens. And he did it. And he showed them. And they all went around and said, yeah, that's right. Okay, we won't believe in that anymore. That's it. It's over. One experiment, that's all he did. And he completely blew that out of the water. Did you see Louis Pasteur upstairs in the lab? You didn't know he was looking down on you?
No.
He's up on the cupboards. You know where you get the racks? He's right there looking down on you. Look for him on Tuesday. So there's a little statue of him on top. I don't know who put it there, but somebody did. He's looking down on you. He continues to do his work. He's watching you, right? All right, now, more recently, we discovered things like restriction enzymes. Back in the 1970s or so. These are very important for today. A lot of the things that we do in biotechnology today, we would not be able to do.
Yeah.
You know that human insulin inside bacteria? How did we get it in there? We had to use restriction enzymes. And restriction enzymes are like little tiny molecular scissors. They're like little scissors of DNA. They cut DNA. So restriction enzymes, they cut DNA. So I can, for example, take a sample of your DNA. Watch this. And let's say this is the insulin gene. I can use these molecular scissors to cut this gene out of your DNA. I'm just taking a sample of your DNA. I'm not taking all of your DNA. Now I have your insulin gene. Okay, then what? Well, I'll cut the DNA from bacteria. I'll open it up. And then I'll put your gene inside here. And I'll close it back up. This is bacterial DNA. And this is your human insulin gene. Then once I close it back up, I will now put it back in bacteria. I will put it back in bacteria. But now it has your human insulin gene. And what will this bacteria make? Human insulin. What do we call all of this? Biotechnology. Who's going to do this? You will, this semester. You're going to make bacteria fluoresce green. Bacteria normally does not fluoresce green, but you're going to make it fluoresce green by doing similar procedure. Yeah.
Is there a specific enzyme? Because if it's cutting,
There are hundreds of different kinds. And they cut based on sequences. So for example, I'll give you an example. Now I'm going back to my research years. There might be a sequence around the gene here that is GAATTC. Oh, I can't believe I remembered it. And let's say another one, GAATTC. That's EcoR1. That's the enzyme, you don't need to know this. That is the enzyme called EcoR1. E. coli restriction enzyme number 1. You see that? So where do we get these restriction enzymes from? From bacteria. Bacteria make these restriction enzymes as a means to protect themselves from viruses that infect them. So there are some phage viruses that infect bacteria. Viruses can infect bacteria too? Yes, we'll talk about it. So what does bacteria do? They use their restriction enzymes as a form of immune system. They don't have antibodies. Bacteria, they don't produce antibodies like we do. They produce restriction enzymes because the only one that threatens them is this phage that injects its DNA in them. So the phage virus, right, the phage infects the bacteria by injecting the phage DNA in the bacteria. So bacteria says, I wish I had something to break the DNA before that DNA hurts me, restriction enzyme. So the discovery of restriction enzymes in bacteria revolutionized the field of biotechnology. Without restriction enzymes, I wouldn't be able to cut the DNA to transfer the gene here to make bacteria make human insulin or growth factor or whatever. So remember this, bacteria produce restriction enzymes to cut DNA, and the main thing that they're trying to use it to protect themselves from is the phage that infects them. Something like a virus that infects only them. They're called bacteriophage. A phage that infects bacteria. Again, bacteria, they're not making these restriction enzymes so we can do this. No, a biotechnologist, after they discovered the restriction enzymes, said, wait a minute, I can use these as a tool in the lab. This is done in a test tube in the lab. You take the DNA from humans, you cut the human insulin gene, okay, I got it. Take the DNA from bacteria, you open it up, and then there's another enzyme that brings them together. Got it, everything's good, yeah, put it in the cell. So it's biotechnology. Now once you have this, you can grow this forever. So I think you get the idea, without restriction enzymes, we'd be so far behind. In the early 80s or so, mid 80s, another major invention, polymerase chain reaction. That's what PCR stands for. Polymerase chain reaction. That's a fancy 3 words for what? A DNA copy machine. There it is. If you leave one molecule of your DNA over here, and I happen to grab it, cotton swab or something, I can now put it in the PCR machine and go like this. Watch, I only put one molecule in there. Put it in the PCR machine and do this. Watch. Copy. Now I have 10 million, then a billion copies, if I want to. So it's a DNA copy machine. There's no lab in the world that calls itself a lab without having at least one of these machines. So huge invention. And it came from the story is this guy who used to do this reaction by hand. He would copy the DNA, and it's very meticulous. Like, you gotta increase the temperature, then decrease the temperature real slow, and then increase it again slow, and then increase it. So up and down, the temperature up and down, up and down. It's called cycling. And then he's going home one time. That's what he mentioned. He said one time. And he's sitting in the car thinking, there's got to be an easier way. I spent too many hours in the lab doing this. It's grueling work. There's got to be an easier way. He went and teamed up with an engineer, and they developed this machine, the PCR. It's literally a machine, little tiny block like that, and it's computerized to cycle the temperature, go up, down, so it does it automatically. I don't know how many years later, he won a Nobel Prize for it. Nice, $1 million. So if you ever see me sitting in my car going like this, I'm trying to win a Nobel Prize. It doesn't work. It doesn't work. Yeah. All right, here's another one. I just mentioned this to my daughter the other day. Small RNA, because she's studying something that has to do with small RNA. And I told her, you know, when I was in college a long time ago, my professor said there are 3 kinds of RNA. Messenger RNA, transfer RNA. Oh, you're waiting for me to say. Messenger RNA, transfer RNA, ribosomal RNA. There you go. And then, this is when I was a student a long time ago. There are all these small pieces of RNA, you guys, yeah? Don't worry about it. They're junk. We don't worry about it. That was my professor saying. So I didn't worry about it. He didn't worry about it, I didn't worry about it. Nowadays, worry about it. They're huge. They play a huge role. Huge role. They regulate gene expression. And it's amazing, the intricate detail of these things. They literally can determine whether or not a specific protein is made or not. Even though you have the mRNA for it. You think you're going to make this protein when you have the mRNA, am I right? Not if you have one of these that says, no, not today. So these small RNAs play a huge role in gene expression. So my professor lied to me, right? But it's because we didn't know back then. Now it's a huge discovery. And you'll learn more and more and more about it. Why? Because now we can use it as therapy. You might actually eventually, if not now, do it. I'm not sure if there's an example of it. I think there is, where you are injecting small RNA in someone for treatment. Then, more and more recently, the Human Microbiome Project, huge project. I think it's still going on. They're trying to map out all the bacteria that live on you and in you. Huge project. And of course, that's going to add a lot of knowledge for us. Now, if we go on this side, this is more medically related discoveries. Real quick, a couple of interesting stories here. Oliver Wendell Holmes, linking infections with mothers who delivered in a hospital. This one here. The Oliver decided or figured out or found out that women in this particular hospital at this time who delivered babies in that hospital never went home. Never went home with their child. Why? Because they both died. They all died. That was his discovery. He looked and see how many women went in to deliver babies and realized that no one is going home. They all died. Then Ignaz, I will not even attempt his last name, decided to figure out what's going on. What he found out by basically watching some of the doctors, he noticed that one doctor, the doctor that's going into the maternity ward,
Never wash their hands.
is coming from the autopsy room. And he followed them one time. Like he said, he didn't wash his hands. That's all it is. So back then they used to do autopsies, no gloves. And then go, oh baby, there you go. They both died. Then came Joseph Lister again, the same Joseph Lister, and said this. I'm just trying to make it as fun as possible, the story, right? he went to the same location and he watched the doctor walk out of the autopsy room and he said, hang on, yeah, wash your hands. Yeah, wash your hands. All right. Maybe they both went home. Simple. So every time you put a pair of gloves on, gown, lab coat, you thank Joseph Lister for that. Because now we have certain protocol. It's not just washing hands.
Yeah.
It's you have to wash your hand, wear gloves, mask, whatever, in between patients, right? God help you if if the hospital that you're working at has a protocol in place where you have to wash your hands, wear gloves, deal with the patient, and then before you leave, you take the gloves off, wash your hands again. If you break that, you run the risk of being fired over it. So obviously we've added layers to this protocol.
I work at a hospital and there's some rooms where you have to take off your, take off your mask and everything in the room.
Yeah.
And there's some rooms where you have to take it out outside.
Oh yeah, every
So
room, every hospital will have its like regulations, even within the different departments.
But it also depends on the patient room
Of-
and what they have,
of
whether
course.
it's airborne or whether it's
Of
contact.
course.
So sometimes
Of course.
you have to read, so it's like, okay, am I taking it out when I leave or do I
Yeah.
have to take it out before I leave?
Yeah, yeah.
So it's very.
And if you break that rule and you get caught, ooh, lights out for your job, right? There are some times when they wouldn't allow visitors in because the visitors for sure are not going to follow these rules. Yeah. Louis Pasteur again. This guy is everywhere. Louis Pasteur. He basically talked about the need for boiling things, and we call that pasteurization. We pasteurize milk. We pasteurize beer. Thank God we cool it down again, right? So we pasteurize things to get rid of living organisms. So anything that's alive, right after pasteurization, they're mostly going to be dead. Most of them. But there are sometimes, there's sometimes what's called endospores. We'll hear about it, we'll study it. Spores are resistant to heat. So even if you pasteurize, the endospore is still there. And that was through the discovery of John Tyndall and Ferdinand Cohen. They found, John Tyndall found that there's a resistant form of bacteria that heat does not kill. And Ferdinand found what they are, the endospores. And we'll talk more about what endospores are. So how do you get rid of those? Autoclave. So we have a machine upstairs called the autoclave. The autoclave comes at the bacteria in 3 ways: heat, pressure, and moisture. Nothing survives that. So the things that come out for us to use, the media plates that we got last week, 2 days ago, they were sterilized. They were autoclaved. That means nothing should be on there other than the food. So that when you open the plate, you introduce only one bacteria on there. Otherwise, it'll be a complete mess when you incubate. So sterile. Beautiful question on the exam. What is the definition of sterile? Sterile is the killing or the absence of all living organisms, including endospores and viruses. That's what sterile means. So if I say to you, this media is sterile, now you know. There's nothing alive in there, and there's no viruses in there, and there's no endospores in there. It's been autoclaved. Joseph Lister, when he said wear gloves, protect yourself, whatever, that's referred to as the aseptic technique. He's the one that introduced the aseptic technique. Protecting yourself from a potential pathogen or contamination. You will learn the aseptic technique Tuesday. Because I need to show you the aseptic technique because you have to use it when you inoculate bacteria from one source into another. Watch this. I'm going to give you a culture too. And I'm going to give you sterile media. Now we know what sterile means. Free of all living organisms, including viruses and endospores. So the only thing that's in there is the media and the air, right? Okay. You want to remove a little bit of my culture and put it inside this tube. Let's say E. coli. But you only want to introduce E. coli in here. That's it. Only E. coli, for example. But you're using a tool, you'll see in the lab, you're using a tool that has a whole bunch of bacteria on it. If you go and dip this tool in here and drag, yeah, you will introduce E. coli, that's for sure. But you'll introduce maybe 10 other bacteria in there. You'll have a contamination. You see? So before you do that, you have to heat that loop red hot under the flame. That's why we have Bunsen burners.
The flame that student.
Yeah. We won't talk about it. So I'll show this to you, where you heat the loop red hot. Now you know for at least that specific time, there's nothing on there. It's sterile. See? That's what you're trying to do. You're trying to use the fire to sterilize the loop. And it works perfectly if you do it right. Then you go in there, take your sample, and right away you go into the other one and release it. So what are you preventing? Using the aseptic technique, you are preventing a contamination. The dragging of unwanted bacteria into a source that you did not intend them to be there.
So I'm confused about the difference between pasteurizing and sterilizing.
Oh, pasteurizing is just basically when you boil something.
Okay.
Like you take, for example, sometimes we get like a water main break and you get a message on your phone, for the next 48 hours, you better boil your water. That's pasteurization. You're killing living organisms. But if there are endospores in there, that heat is not enough to kill the endospores. But it's usually enough for our purposes. Sterilizing is different. Sterilizing is killing the same living organisms, right? But on top of that, you're also getting rid of the endospores and the viruses. Yeah. The final thing here is the germ theory of disease. Again, it was suspected that these microorganisms cause disease, but Robert Koch was one of the first scientists who put in place and confirmed that, microorganisms cause disease. Confirmed. Well, we know that now. And that's called the germ theory of disease. And those are called Robert Koch's postulates. And we'll talk about it in another unit. But he is the first one that confirmed that, microorganisms cause disease. Well, we know that now. Let's quickly, I'm not going to spend too much time on the scientific method. Just real quick. You should know all of this already. And I'm going to more focus on just this part here. The steps of the scientific method, and the reason behind that is anything that we do in science, you must apply the scientific method to. And it definitely, you should know this by now. It starts with observation, observing something. Let's say, I always use this example, the grass is green. Great, observation. You're the scientist, I'm not. When we walk outside to go to lunch, you stop and you ponder, you wonder, you ask a question. Why is the grass green? Great. Me, I'm more worried about lunch, right? So I'll leave you. You want to think about it? You think about it. I'm going to lunch, right? Because you're the scientist. Then as you're standing there, you formulate a hypothesis. If you remember from Gen Bio 1, your hypothesis has to have two things. Number one, it has to be logical.
And falsifiable.
and falsifiable. You have to be able to test it. The hypothesis that you state, you have to be able to go to step 4 with it. If you cannot go to step 4, then all you have is a hypothesis. An example of that. When they initially decided to do the Human Genome Project, that's great. The problem is when they were about to start, the human genome is the sequencing of the human gene. The human DNA. Huge project back in 1990 to 2000. When they first started, they realized something. What is that? We don't have the machines for it. We don't have the computer programs for it. We don't have enough people for it. I think we need to build robots. Why? Because it will take us forever. Otherwise, the robots can work night and day and not complain. You see? And they said, okay, hang on then. Hang on what? Keep it at the point of hypothesis, right? Until we develop the machines. So sometimes you're stuck from going to step 4. Why? Because of the technology. It's not available. But once the technology became available, they went to work. And now we have it. You want to know what your blue eye gene sequence is? Look it up. It's online. Within a second, you'll see the sequence for it. You want to know what the sequence for your catalase gene is? You get it. Any sequence for any human gene, and not only human, we have other organisms as well. Many of them. The other possibility is that you make a hypothesis statement that there's no way you can test. No way. What's an example of that? I always use this as an example. I hypothesize that God exists. Good luck trying to test that one. You can't. You can't go to step 4 with something like that. Why? Because for me to test God exists in this universe, I have to have a duplicate universe, watch this, where God does exist. And we call that the control group. Am I right?
Mm-hmm.
And then I ask the question, okay, compared to that universe, does God exist in this universe? Are you kidding? Everyone's like, what? There's no way that can happen. Of course there's no way it can happen. So now I'm stuck to doing this. I'm stuck to doing this. Watch. Believe me, God exists. But believe me is not science. Believe me is religion. So now we're in the realm of religion. But the problem with science is there's no such thing as believe me. The grass is green because it has a pigment. Believe me, this drug will work, control your diabetes. No. There's show me. Show me the data. You see?
Yep.
You have to be able to go to step 4, to gather the data, to analyze the data, to make conclusions, revisit the hypothesis, and either accept or reject your hypothesis. And then make conclusions. One of the worst things students make, or mistake they make, is that when they write a conclusion, they start the statement, and a lot of us are guilty of this. They start the statement, the conclusion statement, with the 2 words that you should never use. No. The 2 words are, I believe. Whatever. Are you kidding? You went through all of this. You made an experiment. You gathered the data. You analyzed the data. And at the end of all of that, you still want me to believe what you believe? I don't believe anything. You see what I'm saying? You have to show me. So never start a conclusion statement with the words I believe. Ever. The better thing to say is, for example, in conclusion, what up? Or the data shows, what up? But the I believe, you leave that for your religion. You see that? So watch out for that. The final step, which a lot of students miss in a scientific method, is this one. Communication. And without communication, how are you going to know that I have a cure for cancer? How are you going to know that? If I develop a cure for cancer and I put it in the drawer over here and I never share it with you, and then you come 10 years later and figure it out, you say, I say, I had that already. Well, you didn't show us. So this is our bread and butter in research. This is the go. Without communicating how am I going to get grants? How am I going to keep my lab open? How will my boss know what I'm doing and all that? So you understand what I mean? How will I even build a name for myself if no one knows what I'm doing and what I'm getting? This becomes the critical part for your survival in the field as a scientist. Okay, real quick. Again, I need to finish the unit, so I don't know if we'll have time to practice the content. If not, don't worry, we will practice the content. All right. Binomial nomenclature. This is taxonomy. This is the taxonomic categories. This should not be new for you. Should not be new for you. So the most inclusive is the domain. There are 3 domains. Name them.
Bacteria, archaea, and
Bacteria, archaea, and
eukarya.
eukarya. Very good. Bacteria, archaea, eukarya. Where do you belong?
Eukarya?
Eukarya, because you're not bacteria or archaea. You agree?
No.
Where do viruses belong?
Nowhere.
Why?
Because they're not cell.
Nice. Did he know I was going to ask that question? No, I don't think so. But he got the answer right. How do you know? I just know. You probably heard about it before, read about it before, whatever. But the idea is that he remembers it. And that's half of the battle, at least in biology and micro, is remembering things. He's right. Why are viruses not prokaryotes or eukaryotes, or they don't belong in any one of these bacteria, archaea, eukarya domains? Because they're not cells, right? They might have their own naming scheme or mechanism, but they're not going to be under any of these 3 domains. They're not bacteria, they're not archaea, and they're not eukarya. Are they prokaryotes? No. Are they eukaryotes? No. What are they? None of these, because they are acellular. Because when you talk about prokaryotes and eukaryotes, you're talking about cells, and they're acellular. But they do have a naming scheme or mechanism, but is controversial to some extent and not fully worked out, and so we're probably not going to go there. So this is more for the bacteria, the archaea, and the eukarya. The most inclusive is the domain, followed by the kingdom, the phylum or division. You can call it phylum or division. Class, order, family, genus, species. So all the eukaryotes are in the domain eukarya. All the bacteria are in the domain bacteria. All the bacteria that lived, went extinct, all of them are in the domain bacteria. And all the archaea are in archaea. You know, this is similar to you, for example, if you're lucky enough to have a walk-in closet at home, but your walk-in closet is huge. And it's very organized. And all of your clothes is in that closet. Anything that you think is clothes is in there. Your purses, your shoes, your belts, everything, right? So when you're standing outside of this closet, here's what you're going to do. You're going to do, this is my domain of clothes. That's domain. All my clothes are in there. All bacteria are in domain bacteria. All archaea are in the domain archaea, and so forth. But you are very organized. So you have shelving units. The first shelf is the kingdom. The kingdom of what? The kingdom of pants. Are you catching it?
Mm-hmm.
That's the shelving unit. The third shelf is the phylum. The phylum of what? The phylum of genes. You getting that? The second stack are what? Black color, whatever, right? So this is the black genes, and so forth. So you're going from the most inclusive, and you're homing in on the one pair of jeans that you'll be wearing that day. And what would that be? The species. So you're going from the most inclusive to the Least inclusive. When we get to this level, every organism has a unique two-name designation. That's what's called a binomial nomenclature. Binomial, two names. The genus followed by the species. And they have to be written in a very specific way. All letters are italicized. This is for humans. All letters are italicized. The genus before the species, and a space in between. Just like you see here. The first letter of the genus is capitalized. So if you have an italics on your computer, and we all do, if you are typing, it is expected that you do it this way. But if your italics is broken, then you must underline. No italicize, but you must underline. So you can either italicize or underline, but you can't do both. You can abbreviate as well, like this. You usually abbreviate using the first letter of the genus, but you have to write the full name of the species. If you are handwriting, you must underline. Why? Because I can't tell you if you're italicizing or not. But I have very good handwriting. I don't care. You have to underline. I know of one of my friends who wrote a paper, a research paper, and sent it for publication to get it accepted. His data is awesome. It was rejected because he didn't write these the right way. To our review panel, because it gets reviewed, they look at this thing and they go, ooh, he doesn't even know that? I don't trust the data. You catching their point of view? If he doesn't even know how to do that, or if he's too lazy to do that, I don't trust his data. So it got thrown back in his face. That's terrible, but oh. So you have to follow these things. That's a nice question on the next exam. Final slide. You don't need to know this thing. I'm talking about this. You don't need to know that. All right? This is a quick discussion on evolution. We already talked about we all came from a common ancestor, and then through evolution, which is also referred to as descent with modification. This is descent. Descent means going this way. We're going from one time point to the more recent time points. This is descent with modification. What's the modification? Genetic mutation. Through time, we acquire genetic mutations that are either beneficial, harmful, or do nothing to us. Those are the three types of mutations. Silent mutations are the ones that don't do anything to you. They just mark you for who you are. That's it. The harmful ones, those are the ones that cause disease in you and I. And then the other ones that natural selection uses to select whether you survive or not. Survive what? A specific environmental change. None of this can be controlled. You cannot direct your evolutionary path. Can't direct it. For many reasons. Number one, you can't control the environment. And the other thing is, you can't control genetic mutation. Genetic mutation is a random event. This is called a phylogenetic tree. The construction of some kind of tree-looking piece of information that shows the interconnectedness of all living organisms, and not even living extinct. If it flew in the air, if it walked on land, if it dug in our soil and swam in our waters, it will be on the tree of life. Taxonomists, that's what they do. When we discover a new species, that's when they go back to work, trying to name it, trying to classify it, and put it on somewhere. Where does it fit on the tree of life? This new thing that we discovered. So that's a quick cover of evolution. And so, any questions? I know we talked a lot. I just wanted to finish the unit so I can send you home.